Distinguished guests, ladies and gentlemen, and my dear guests online and offline, and good morning. And I come from Maidri. I'm in charge of the ISO15189 and the lab medical certification. And you can say that in December of 2021, we organized the first session. In May last year, we organized the second. Today we're going to have the third episode of this ISO15189 medical lab quality management. The series of these episodes are committed in discussing the cutting edge topics in the ISO 15189 medical lab quality management. Today, we're going to have the third episode. We're going to talk about lab individual quality control based on risk management. For this session, we're much honored to work with our Chengdu University of TCM to organize this episode. So coming next, let's welcome Professor Qiong Daqian from Chengdu University of TCM for the opening remarks, please. Distinguished guests, ladies and gentlemen, my dear colleagues, good morning. I'm Professor Qiong Daqian. I'm the Director of Clinical Laboratory Department of Chengdu University of TCM. I'm very honored to be the president of this symposium. Thank you very much to spare your time to sit down with us together for this symposium. In order to better improve our methodology and practice of medical lab management, continue to strengthen operation with our international peers. Today, we'd like to organize this hybrid symposium with online and offline participants being here to jointly talk about ISO 15189 medical lab quality management. We have one plenary session in Chengdu, Nanning, Kunming, Chongqing as subsites. In Chongqing, we are very happy to have Professor Deng Kun, Director of Clinical Lab of the third affiliated hospital of CQMU, to be the president in Chongqing session. Professor Deng, please say hello to everyone. Thank you. We're in Nanning subsession. We have the Director of Clinical Lab of the second affiliated hospital of Guangxi Medical University, Professor Xie Li, to be the chairman of the Guangxi session. We're in Yunnan session. We're very happy to have Professor Chai Lin, Director of Clinical Lab of Kunming Municipal Hospital of Traditional Chinese Medicine, to be the chairman. And we're also very happy to have Professor Hong Ning, Director of Laboratory Department of Qianxian People's Hospital, to be the chairman of that session. We're very happy to have our honored guests. They are Professor Wan Nan, the Vice Director of Clinical Lab Department of General Hospital of Northern Theater Command, President-elect of CRSI, Professor James Nicholas. Ladies and gentlemen, I'd like to thank you again. Thanks for all your support, and thanks for all the guests of being here with us today for this session. I specifically would like to thank Maidri in supporting us for this symposium. Last but not least, I wish a complete success of this meeting. Thank you. Thanks for Professor Xiong. Thanks for your opening remarks. And I surely believe we're going to have a more improvement and mutual learning from each other. Maidri attaches great importance for this session. Coming next, let's welcome Mr. Wang Peng from Maidri to give a few words. Professor Xiong, distinguished guests, and our clinical expert and clinical lab expert, and Professor Nicholas. It is my great honor to be here on behalf of Maidri to talk to many of you. We know that since the year of 2030, ISO 15189 has already become a key topic in our industry, especially it's played a vital role for the quality management system building for any hospital. And we also clearly notice, especially in the southeast part of China, and especially where our hospitals been located nowadays, we have 94 hospitals that has been certified by ISO 15189, and I surely believe in the near future, we're going to have more hospitals to be certified by ISO 15189, which will lay a very solid foundation for our future development. So, as a representative from Maitri, we're going to step up our efforts and to support the hospitals to be certified by ISO 15189. For Maitri, we're going to do the following things. First of all, we're going to improve our product to provide high quality solution to all of you, including our new generation product, the CBR Spectrum platform. We leverage the microwave cleaning in order to get more precise test result. Besides the high quality product, and we also started to organize our supporting team and to our lab expert. Hope that we will be able to provide you the consultation service. In that way, we can help the hospitals to build your quality management system respectively. Well, with the reopening policies being available in China, we're also going to organize more events like what we have today here. We have Chengdu as the plenary session, Guangxi, Chongqing, Guizhou, and Yunnan as the satellite session. We're going to talk to each other to further strengthen our cooperation. We also would like to engage more international experts to share the international experience with our colleagues sitting here, and especially the ISO 15189 certification and how we're going to do the risk management. Last but not least, I wish a complete success of this session. Thank you very much. Thank you very much. Thanks for Professor Wang for your nice remarks. And I also would like to thank for all of you sitting here. And thanks for supporting my dream. And coming next, we'd like to get into the presentation session for today. First of all, please allow me to introduce our first speaker. Our first speaker is Professor Deng Kun. And Professor Deng Kun is MD Professor, Chief Technician, and Doctoral Supervisor, who is also the Chongqing Young and Mid-Aged High Talents, and who actually published multiple articles in multiple journals, and who also finished and chaired many very important scientific projects. And altogether, he obtained 3 innovation patents, where at the same time, Professor Deng Kun's been playing very important role in multiple academic associations. Coming next, let's give the floor to Professor Deng Kun for the presentation, please. It is my great honor. Thanks for the nice introduction made by Professor Xiao, and thanks for the invitation made by Professor Xiong. With Professor Xiong's great support, we have already organized multiple discussions and meetings regarding ISO 15189. Please allow me to introduce our keynote presenters for today.
Hello, Professor Liko. Can you hear me?
Yes, I can hear you.
It's my great pleasure to meet you at the webinar. Of course, we prefer you to come to China and Chongqing in the future. It's my great honor to introduce you to my colleagues. Come from Chongqing and Kunming and Guangxi province and Chengdu province. So my colleague, I will introduce you in Chinese to my colleagues. So, okay. 刚才我已经跟
Okay, great. So just now, I was talking to Professor James. So coming next, please allow me to introduce Professor Nicholas. Professor Nicholas is actually president-elect for the RSI, who is also the medical director of clinical chemistry and the point-of-care test of Vandenberg University, and who is actually working as the Director of Clinical Chemistry and Real-Time Laboratory Medicine of Vandenberg Medical Center, and who also is a researcher of the clinical chemistry postdoctoral training program of Mayo Medical Center, and who used to work for Johns Hopkins as the chemistry lab vice director, and who is actually very famous in this industry, who had wide experience here, and he's truly expertise on evidence-based chemistry testing and also toxicology. And Nicholas used to also work for CRSI for multiple positions, and who is also the member of the CRSI and also the president of the real-time testing consensus community. And today, I think he's going to mention a lot about POCT, and we'll also be very happy to have Nicholas to be here. He actually published, he'll actually be the president of the Protocol Assessment Expert Committee.
Aware of your professional background and achievement. We are looking forward to your presentation very much. I'm going to turn it over to you. Okay, thank you.
Thank you so much. I am honored by that introduction. And I do want to thank the Chengdu University of Traditional Chinese Medicine for organizing this session, and for Mindray for supporting this talk and presentation. I'm going to share my screen now. 1 second. Takes a second for it to load. And there we go. Can you see that okay?
Yes, we can. Okay.
Perfect. Let me get it to the beginning. There we go. Thank you so much again for this invitation. And as was introduced, I'm professor of pathology, microbiology, and immunology, and medical director of clinical chemistry and point of care testing for Vanderbilt University School of Medicine, and medical director in clinical chemistry. The objectives of today's talk are to recognize common sources of laboratory error, to identify the Clinical and Laboratory Standards Institute, CLSI, document EP3 as a resource for risk management, and to explore the variety of control processes that manufacturers utilize with laboratory devices, and to describe the benefits and challenges of implementing EP3. Now, when we talk about quality control of laboratory tests in the United States, under our Clinical Laboratory Improvement Amendments of 1988, that requires and mandates that every laboratory test that we conduct runs 2 concentrations or 2 levels of quality control each day of testing. But outside the United States, ISO15189 requires that laboratories shall implement quality control procedures for individualized quality control and internal and external quality assessment to verify that the performance of the examination meets the clinical needs of their patients. ISO 15189 also requires acceptance and rejection criteria set up to indicate when the performance of that examination isn't sufficient to ensure the consistent validity of results. It should be, these acceptance and rejection criteria should be defined and judged in relationship during the actual verification and validation of the method. The frequency, the measure end concentrations, and the result types of internal quality control program for each examination procedure should be defined based on the stability of the procedure and risk of harm to the patient. Now, I could talk about instrumentation and automated equipment in the core laboratories that do high volume testing, but I thought I'd simplify this talk by using point of care testing devices. They're very simple. They're intended to use by non-laboratorians with very minimal background and training, and they oftentimes use single test cartridges. So when we run quality control on a unit use laboratory cartridge, it consumes that entire test or cartridge with no guarantee that the next test is going to perform the same. So these unit use devices have been manufactured and engineered with built-in biologic, chemical, and reagent control processes that manage the quality and reliability of each and every test. In many instances, when we run two levels or two concentrations of liquid QC on these unit use tests, it duplicates what's built inside that cartridge by the manufacturer. We tried to mesh this with the government mandate for two levels of liquid QC each day of testing. And to do that, we brought together a committee of experts by CLSI and defined a document, EP3, that will provide laboratories a means of defining internal quality control programs for managing the risk of this test. The U.S. Centers for Medicaid and Medicare Services adopted the principles of CLSI EP23 as individualized quality control plans. That's the name that was given to it by CMS. This is the actual document. It is currently under revision and should be released later this year in edition 2. It is Clinical and Laboratory Standards Institute EP23A, it's approved, and it is laboratory quality control based on risk management. Now, under the CMS Individualized Quality Control Plan, defined by EP23, EP23 really introduces industrial and risk management principles to the clinical laboratory. The Individualized Quality Control Plan, defined by CMS, allows laboratories to develop a plan that optimizes the use of internal built-in and engineered control processes on that device and balance it with the cost and resources of having to analyze liquid quality control without impacting patient safety or the reliability of that test. And CMS adopted the EP23 risk management concepts into what they call an individualized quality control plan. Under that option, laboratories have two ways that they could go. They could measure two levels of liquid quality control each day of testing, or for some tests like blood gas testing that are especially risky, they required 3 levels each day of testing, as well as hematology tests. Or a laboratory could take the pathway of developing an individualized quality control plan that balances internal control processes with running these liquid external controls. It allows us to reduce the frequency of analysis of liquid QC to the minimum recommended by the manufacturer. And it maximizes clinical outcome, saves staff resources, and is cost effective to the laboratory. There are 3 parts to developing an individualized quality control plan. The first is a risk assessment, where we ask, what could possibly go wrong with this test? Let's follow the sample through the testing process and look for weaknesses in each step of that process. For each weakness we define, a hazard, we come up with a way that we're going to handle that. And that's defined in our quality control plan, a control process for that risk. And then we monitor, once we implement our quality control plan, monitor for continued errors or failures. That's the quality assessment. And when we see a continued problem, we go back and reassess our risk, modify our control plan, and implement a revised plan. So it's a continuous quality improvement process. That's the three components of an individualized quality control plan. So let's ask about risk in the laboratory. And risk is a little bit of common sense. We are all laboratorians. We have a lot of experience in pre-analytic evaluation of tests and errors that can happen before a sample comes into the laboratory. And I think we all, with our common sense, know if this person starts up that chainsaw in this way, this is not going to end well. So we start with risk by saying that nothing is perfect. There is no perfect laboratory device. Otherwise, we would all be using it. Any test can and it will fail if we use it under the right, or what I'd like to say, the wrong conditions. So a discussion of lab errors really has to start with what could go wrong with the test. We map our processes and identify weak steps during testing. We have to understand what are the sources of those errors, those hazards in our testing process. And for each hazard, we mitigate that risk by preventing a result either before the lab reports it or detects it and contacts the clinician before they take clinical action. Essentially, we have to conclude that lab tests are not foolproof, and we have to make them as foolproof as possible by implementing control processes. Sometimes it's staff training, enhanced maintenance on the device. It may be other functionalities. And I'll give an example with a molecular test. Here's the pathway for risk management of EP23. First, we create a process map. Follow the sample, pre-analytic to analysis to posting of the results and what happens after analysis. Identify weaknesses in each step of the process. And for each of those weaknesses, identify how we're going to mitigate that risk. What are we going to do to prevent that problem from happening in the testing process? Then we're going to summarize all of those mitigation steps, those quality processes that we put in place in our quality control plan. As we develop this process map, look for weaknesses in 5 key areas. Samples, issues like bubbles, clots, lipemia, hemolysis, clotting of the sample. To operator, their training, their competency, the stress that they're under in terms of work conditions. The reagents, degradation of reagents during shipping before we receive them in the laboratory or during storage, use past their expiration date, both for reagents as well as quality control, and calibrators. Look to the laboratory environment, dust, temperature fluctuations, heat, humidity, and electrical continuity, water quality, pressure. And finally, the measuring system itself. What happens if there's a software failure, optics drifts, electrical instability, or dirty optics, scratches, and contamination? So let's turn to an example now. I'm going to pick on molecular infectious disease because during the COVID pandemic, a lot of our point of care clinics, community practices implemented molecular disease testing for COVID and for flu. Many of these devices are simple. They're intended to be run by non-laboratorians. They use single-use cartridges that provide no carryover between samples. They're connected to our wireless network. They test for a variety of different pathogens, including sexually transmitted diseases, respiratory viruses like COVID, flu, RSV. They can test for methicillin-resistant staph infections as well as C. difficile. They're portable. They require minimal education for operation. And in the United States, they are classified under the most simple of devices, CLIA waived. They're intended for use in the clinic, other healthcare settings, in the community, outside of the hospital laboratory. They are used in developing countries, emerging markets, military deployment, and they can even be used in disaster relief after tornadoes, hurricanes, earthquakes. When we think of molecular diagnostics as a laboratorian and the way I was trained, we always had unidirectional workflow. We set up our laboratory so that we provided sample, DNA or nucleic acid preparation in one area of the laboratory and move those in one direction to amplification and detection. We didn't go backwards because we didn't want to cross-contaminate samples with the million amplification of all these amplicons. But with unit use devices, we're not presented with this complexity of laboratory settings. The device itself accepts one sample, it preps the sample, extracts the nucleic acid, and amplifies it all within a self-contained cartridge. As with laboratory testing, we are concerned about environmental contamination. Contaminations on pipettes, on touching the sample outside of the sample container, and cross-contamination. Because amplification and molecular diagnostics are very highly sensitive techniques. Very small amounts of nucleic acid are amplified millions fold. So small amounts of contamination can actually cross-contaminate samples and cause false positive results. I show this study, which looked at technologists, and they had them touch a powder on their gloves and then conduct the test. And you can see with under UV light that there was a lot of cross-contamination of this across the environment. So our concern is always, what's going to happen now when we put these molecular diagnostics out in clinics and allow nursing staff and non-laboratorians to conduct the test? In the environment, viruses, fomites, they can persist on surfaces and protect the nucleic acid. We have to teach staff of the common sources of environmental contamination. And one of the risks of environmental contamination, the mitigation step that we put into place when we implemented this, is to require that staff handle only one sample at a time. They clean the counters and the device after testing, and they change their gloves between each sample. The manufacturer makes it easy because they package one cartridge at a time. They have an integrated disposable pipette for transferring the liquid from the sample into the cartridge. And we don't allow them to place the swabs, contaminate the counters with the liquid. We are concerned about carryover, but because of single tube sampling, the reaction being contained in a single-use cartridge, we train the operators not to handle the swab itself, handle the sample on the swab part of this, because that can contaminate their gloves and then contaminate the next. We also train them not to crush the cartridges that contain all the amplicons after testing. So don't put them in a trash compactor. Make sure that they are incinerated or that they are disposed of so that there is not the possibility of contaminating that clinic environment. Manufacturers make this very easy. They have color-coded sample that connects in with the amplification cartridge. This really helps in terms of prevention of environmental contamination and cross-contamination. We also have to be concerned that we have adequate volume. We collected an adequate sample on that patient. There are internal and procedural controls with each assay that flag if the reaction does not occur appropriately. A nasopharyngeal swab is included with each test kit. This ensures that we collect sufficient sample. It includes transport media to ensure that the appropriate swabs placed in the appropriate media, and the swab snaps off to prevent environmental contamination. And in tube sampling, there's an integrated pipette to protect environmental contamination and ensure we get adequate volume applied to the test cartridge. The reagent controls themselves are performed on each and every cartridge, and they're built in. The sample processing, the sample adequacy controls. One is exogenous nucleic acid that's added to the cartridge, and the other is an endogenous that comes from the patient sample themselves to make sure that we had adequate sample. They both co-extract with the amplification of other nucleic acids in the sample. This ensures that we have sufficiency of sample collection, effectiveness of onboard sample processing or application, integrity of the extracted nucleic acid, that it hasn't degraded, and that the reaction occurred under favorable conditions, that there was absence of amplification reaction inhibitors. There's then also software controls that relate algorithms to verify baseline correction, amplification cutoffs, and reduce the frequency of unusual amplification curves. We also have, as we think about sample errors, patient considerations, because giving a positive result, the clinician has to understand where that result came from. Molecular diagnostics should aid in the care of the patient. The test shouldn't define the patient themselves. And the test is only as good as the clinician who uses that test. With strep A, MRSA, methicillin-resistant staph, they may detect carriers that don't have active infections ongoing or may have resolving infections. We should assess clinical symptoms always in conjunction with positive test results. If the patient doesn't have symptoms, but they turn up positive, we have to ask what's happening with this test. And just giving antibiotics to a patient who turns positive is not part of antibiotic stewardship, particularly if the patient doesn't have clinical symptoms. Patients can be carriers and not symptomatic. We also have to think about operators, training, and competency. Fortunately, these simple little devices that are intended for point-of-care testing have operator lockout features. This functions through a number code or an operator ID, identification. There is a list of operators, training, and competency dates that are maintained in the data management system of that point-of-care testing device, such that if the operator walks up to a point-of-care testing device and intends to conduct a patient test, but their recertification, their training has expired, they can warn the operators that they're impending a month prior to expiration, and they can lock them out from testing once they have expired. This really helps support our standardized training protocols, our checklist, and we ensure that all operators get the same information and are trained to the same standards across the board. So this operator lockout supports our standardized training program and documentation. We also have to ensure that operators perform quality control, liquid quality control analysis when they are required at the required frequency. We have quality control lockout features on these devices. They lock up if quality control has not passed and is successful at the frequency we define, whether that's daily, weekly, monthly. Operators can try to get around this by running quality control in the patient mode, but our devices are smarter now, and they can detect the difference between a stabilized quality control sample and a patient sample. and lock out the operator from testing until controls are successful. Operator errors can also occur with patient identification. We need to ensure positive patient ID, use of two unique identifiers that match the specimen to that patient. In other words, patient name and birth date or some other identifier. Because if we chart results to the wrong patient, this can lead to inappropriate medical decisions and inappropriate treatment. We also have the possibility of mixing up samples in the laboratory, misaliquoting, putting the wrong sample on the wrong device under the wrong patient's name. But this risk is reduced if we barcode our samples, and we handle only one sample at a time. So the residual risk is there, even with barcoded specimens, because you can mix up the barcodes during sample collection. And we have seen that happen. It's a low frequency, but it does happen. And we have to train our operators to be aware of this problem. Another operator error can occur during analysis. Fortunately, many of these simple devices are very intuitive. The manufacturer has engineered a simple process to perform analysis. And there's not a lot that the operator can intervene with. The shape of the cartridge is a lock and key fit so that the sample side of it locks with the reagent side. If you incorrectly insert the cartridge, it will not read the barcode and the device won't start testing. There are also color-coded sample amplification cartridges that snap together for analysis. The really key thing of this is that the result is not time dependent. They don't require the operator to sit there and time for 15 or 20 minutes to read a line like on an antigen test. It's automated. It's read shortly after test completion, and you get a printout that can be read later at the convenience of the operator when they come back. We do have to be concerned about reagents and use of expired reagents. Our Centers for Disease Control in Atlanta and the US Food and Drug Administration for glucose testing and for other point-of-care devices have warned that you should check and record the expiration dates of reagents and be sure to discard any reagents or tests that have expired. Fortunately for point-of-care molecular diagnostics, the cartridges are barcoded with an embedded expiration date. So if the operator tries to insert an expired cartridge, it will lock up and will not allow testing. The cartridge is also barcoded with a lot number that automatically calibrates that test. And it will not allow testing until the institution has checked in that lot for patient testing. The barcode also restricts multiple uses of the cartridge because once it's seen a cartridge, you can't reinsert that cartridge and try to do another test. It won't work and it won't read it. It errors for the operator. Finally, we need to think of environmental errors, temperature. Cartridges during shipment as well as storage in the laboratory, they may not sense overheating or freezing during shipment. That's why the internal control processes are so important. They validate that the amplification enzymes are still viable at the time of testing. It's also important that on receipt of new shipments, that laboratories, clinics, the settings where this test is going to be performed that are storing reagents, analyze liquid, positive, and negative controls to verify the reactivity of those reagents upon receipt of that shipment. The instrument, fortunately, monitors environmental temperature. So if the environment at the time of analysis is too hot or too cold, it will lock up, error, and not allow the test to proceed. We can have device errors, but fortunately a self-test is conducted to make sure the device is working when you turn it on, and that there's readiness of mechanical parts for thermal cycling, for detection of optics, the printer, the network, and its connectivity are all functioning. The ambient temperatures of the modules and the cartridges are monitored to ensure appropriate operational temperatures during analysis. And the device will flag an error when an internal malfunction is detected. It also verifies that the cartridge is loaded properly and the integrity of the barcode during loading. I kind of want to end by mentioning the difference between molecular and antigen testing. Antigen-based flu and influenza tests require annual updates, and our Centers for Disease Control require manufacturers to challenge their tests with the new strains of flu that come out each year. This is called flu reclassification, and it's highlighted the limitations of antigen testing to physicians and physician office laboratories. But with molecular diagnostics, this is not a requirement, because the amplification sequences are in the conserved portions of those viruses. Nucleic acid amplification testing, NAT testing, is the optimum diagnostic modality for flu in our clinical practice guidelines. And there's no need to confirm rapid strep A testing, the antigen testing, because you can treat definitively based on the molecular test. I want to conclude by mentioning that we need to develop a process map that looks for weaknesses in 5 key areas: the samples, the operators, the reagents, the laboratory environment, and the measuring system itself. What have we learned at Vanderbilt from our individualized quality control plans after we've implemented them? What we've noted is that there's been inconsistencies in operator technique between different nursing units. For blood gas testing, for instance, some barcoded the sample at the patient's bedside when they collected the sample. Others, however, took that tube down the hall to the analyzer and barcoded it at the analyzer itself. This presents a risk of sample mix-up with an unlabeled specimen. Our IQCP supported the rationale for why we run liquid QC, and it minimized the staffing resources required to support the test, because each of our control actions was linked to a specific hazard in our process map. It essentially gave meaning for why we were doing what we were doing at the frequency we were analyzing controls, rather than meeting a government regulation mandating 2 levels of QC every day of testing. It gave us an opportunity to improve our operational efficiency. Before we did an IQCP, we were running quality control on every I-step rather than quality controlling the lots of cartridges, which was actually the chemistry of the reaction rather than the electronic reader. We started running 3 levels of quality control, every receipt of every shipment, and that met linearity. So we met, and we were running continuous linearities on these, and we didn't have to run a 6-month linearity check as mandated by CLIA. So before, when we were quality controlling the devices, we ran about 716 tests total. Afterwards, we went down from 716 to 304 tests, 388 if we had multiple lots of reagents on hand. So we cut the number of quality controls in half without compromising patient care. There was a considerable resource savings and cost and expenditure of the controls themselves. I want to conclude by showing you some resources. There is EP3 that I showed earlier, as well as an implementation workbook that is available from CLSI. There are ISO guidelines on risk management, ISO 31000. There's ISO 22367, which is the application of risk management to medical laboratories. There's 22583, which is the guidance for supervisors and operators of point-of-care testing devices. And ISO 17025, which is general requirements for the competence of testing and calibration laboratories that introduces risk management to the clinical lab. I also have clinical chemistry, which focuses on real-world examples of errors and includes a chapter on point-of-care testing. Now, I mentioned that CLSI EP23 is being revised. Some things you can expect in the next revision is better alignment of EP23 with ISO 22367 that I just mentioned, as well as ISO 14971. We also incorporate detectability in the risk assessment. So rather than just harm and the risk to the patient, we're looking at detectability by the manufacturer that's built in that device. And we replaced some of the examples, the glucose example, with some real world examples of quality control for non-instrumented single-use devices, instrumented devices, and exempt microbiological media, as well as updating the references. In summary, there are many sources of laboratory error. I have focused on 5 key areas. Risk management allows you to get a better assessment of what you're doing in the laboratory by mapping your processes, your workflow for weaknesses, and allows labs to take action before errors are released to the clinician and medical action is taken. It's an opportunity for laboratories to trace your workflow and reduce risk in the testing process. Remember, manufacturers have engineered a number of internal control processes that help laboratories and operators detect and prevent common errors. Understand, utilize these features to minimize risk of errors in your environment. I want to thank you for your attention, and I'll turn it back over to our host.
Okay, thank you, Professor Lucas. Thank you very much for your wonderful presentation, and your presentation makes us very clearly to know how important it is for patient and how helpful for our clinical laboratories to use IQCP based on risk management. So I was also impressed by your idea talk about which is the good quality management is not IQC and EQC, even not the government regulation, but the patient safety. Therefore, implementation of the IQPC is very reasonable. Thank you very much.
Thank you.
And maybe my colleagues have some questions for you. Could you please just wait a second?
Sure.
我们各位的会场是否有
All right, let's open up the floor and check whether we've got any questions for James.
这是一个很好的机会。
This is a very good opportunity to interact with the professor. Hello, James.
Yes.
Yes, for our academic exchange today.
OK， 后面的答疑是吧？好。
Yeah, we may have a panel discussion in the end.
Professor Nick, thank you very much for your presentation. I think they want to take the questions. They prefer take the the questions to the American tenacity for you, okay? And thank you for your speech today, and look forward to see you in China. And thank you.
Thank you.
好的，那么我们 next 下一个环节是
Right. Next, I'm going to introduce to you the moderator for the next session. The moderator comes from Guangxi Medical University number 2 Hospital, Professor Xie Li, an MD, the Chief Technician, the Master Advisor, Chief of a Lab Testing Department, the Guangxi Medical University number 2 Hospital, visiting scholar to Harvard University, the United States. Currently, she serves as the Youth Committee member of the Chinese Medical Association, the Lab Testing Branch, and the China Medical Clinician Association, the Lab Testing Branch, Youth Committee member, etc. And she took part in 6 national level research projects and chair 3 province level research projects, published over 20 papers, etc. And Prof. Xie is my good friend at the Youth Committee of a Chinese Medical Association. Without further ado, over to you, Prof. Xie.
非常感谢这个邓坤教授的介绍啊，好久不见了。
Thank you very much, Prof. Deng. Long time no see. I'd like to extend my thanks to the organizer for inviting me over so that I can chair this session. The speaker will be Professor Xiong Daqian. First, brief introduction of Prof. Xiong. Prof. Xiong is actually the chief of a laboratory department and a chief of the technician department of Chengdu University of TCM Hospital, and the technical reviewer for ISO 15189, and a deputy committee chief of a Sichuan Provincial Medical Association Laboratory Testing Branch, and China Geriatric Medical Society, the branch of a lab testing, the standing committee member, etc. So Prof. Xiong is working on medical laboratory QC and quality management, the gut microbiota, the cardiovascular diseases, the diabetes, biomarkers, and a pathogenesis mechanism research. So the title of the talk today is the application of quality indicators for continuous improvement. Now let's welcome Prof. Xiong. Thank you for your introduction. So the title of my talk today is Quality Goals, Quality Principle, and the Application of Quality Indicators and a Continuous Improvement. In the process of our operations, when we apply the quality goals and the quality indicators, there are those issues we want to clarify. So I'd like to share with you my thoughts from the following 3 perspectives. First, quality principle, quality goal, quality indicators, and quality requirements. So let's look at the first part first. Basically speaking, the service capacity of a lab is very important, and the number of testing items we assume. Second, the accuracy of the testing results. Third, the TAT. How can we achieve these competency items? It is a must that we establish quality managerial system. We need to be guided by the overall principles for quality. We need to come up with those quality goals and assess whether these goals are attained. So we have those metrics and indicators for quality. And there are quality requirements we need to meet. For quality principles, I'm sure we're very familiar with these principles already. And this is like a framework for drafting quality goals. You look at CNAS 002 2012 version, there's description of a quantity goals and planning. So in terms of these goals, what matters is that we need to make sure that these goals are highly aligned with the principles. And how can we achieve those quality management goals? That is to say, we need to clearly describe the quality goals as a means to interpret the quality principles. So in terms of a service capabilities and in terms of a quality, we need to have those quality goals that we stipulate and with good commitment, that in order to measure whether these goals are attained, then we need indicators. So for the entire testing process, we need monitoring. We need those parameters. For example, the internal quality control, external control, and before the testing, and all these indicators we need to cover. But personally speaking, the quality requirements are very important as well. It is a must that we meet these quality requirements. So there are quality goals and quality requirements here, but in terms of the basic concept, there are some differences. So for quality goals, we may be able to achieve only 80% or 90% of these goals. However, there are minimum quality requirements we need to be able to achieve 100%. So this is the conceptual difference between quality goals and quality requirements. So this slide is only for your information. This is not like the right answer or only answer, but this is our perspective. We need to come up with those specific quality goals as described in the center of the slide. Moreover, for the entire testing process, there are main quality indicators before, during, and after the testing. We need to extract those indicators and come up with a selected list. So these are some of my personal thoughts, but I'm sure it takes no explanation for these basic concepts. For example, quality improvement, quality planning, and QC. For planning, for example, it is a part of a quality management. Which type of requirements shall we come up with respect to effectiveness and efficiency, and how these are related to quality? Taking effectiveness as example, the extent to which those planned actions are delivered, and there is audit and review, and to carry out objective assessment based on objective evidence. And from national authorities, there are some of those documents. So second, let's talk about the change of the quality indicators and how they have been applied. As we know that Article 4.14.7, this is a section about quality indicators, mentioning it is a net must that we assess the effectiveness of these indicators on a regular basis. And this is a 2015 edition of the clinical testing medical care quality indicators. There is a 2017 edition as well. For 2017 edition, it includes all those items available as per 2015. But what's changed? In 2017, there are some new descriptions which are different from 2015. Some of those contents are deleted, especially some term. For example, the non-conforming rate. This word has been deleted from the 2017 edition. For 1517, the two editions, 15 edition and 17 edition, there is a difference in terms of the number of those indicators before, during, and after testing, but the main framework is pretty much consistent. So there are those quality indicators before testing, during testing, and after testing. There are 3 indicators for the process quality indicators. I think these 3 indicators are very important.
We also attach great importance to the following 3 criteria, and also in the appendix, it is actually a more value-added information that is being highlighted in the, for example, TAT and also other proteins. But let's also take a look at what has been mentioned by the National Health Commission regarding the 2090 newly added criteria being built into the AAA hospital performance evaluation. For example, you can see that in 2020, and for the AA hospital, they also have a newly added criteria for the medical lab to be complied with. And you can also see that how can we actually leverage lab quality criteria to further improve our quality management, that is PPCA. And this is a very important step. We need to actually do very good planning, operation, identify the gap, and then to make further improvement. First of all, we need to do very good planning, and we need to clearly understand what would be the lab management quality criteria. Regarding the quality criteria or quality standard, some of the people said, let's just build the quality criteria or the standard indications into the document. But you need to clearly tell people and how you're going to monitor the quality indication and especially how to evaluate the quality indication, whether it's been reached or not. If not yet been reached, and you need to find out the countermeasure solution. For example, and we're just using a table to make the real-time recording of our tests. And at every quarter or every month, if we have the review meeting, we can use a table to help to understand the situation of our instruments. You can see this is the queuing time for blood collection in 2020 in H1 for the outpatient clinic. And therefore, through such a table, we can clearly understand what are those improvements we could make in order to help to shorten the queue time. You can say that we also pay much attention to the 75 percentile and the 90 percentile in order to further improve the service. And sometimes the data difference in different months is also due to the blood collection volume difference. This is actually the periodical review, and we can actually take different criteria to understand how the lab operations been done. There are a few very important indications that we need to pay special attention to. There are a few indications that are not going to be performed each month or each week. And you know that for periodical review, we need to make sure that we have good quality management, internal review, and the medical assessment, managerial assessment and review. Those are very important indications. Right after the test, we should also make sure that the documentation has been well done. And we need to also make sure that our indication be effective throughout the journey. In such a way, we can propose the following improvement in the past. And we'd like to understand whether our indications being effective, whether there's any gaps being identified. For example, like Professor Nicholas mentioned, we can leverage the sample, the reagents, environmental factors, and instruments. We need to pay attention to each of the factors, assess it one by one, and then to identify the place that we can make great improvement. Let me also talk about the third point, also the final point of my presentation. I actually have 2 indications that can help to improve the quality of my lab. The first one is still planning. For CV, what should we do? I used to encounter some of the troubles. Maybe I'm not 100% correct. In 2050, you can say that we mentioned for the lab quality management, and you know that the loss rate, and they need to be greatly improved. So that's a reason we need to think about it. And for example, for the first criteria, we need to be clearly understand. And if the annual failed rate is less than 5%, then some lab believe they actually be able to have a very good result. But we have to think about it again, and we actually make sure that we subbar to some of the criteria. And if our failed rate or we are subbar or unable to meet the criteria, what we can do? For each methodology, we need to make sure we need to be 100% comply with the CV requirement. This is actually for the chemistry project variants. And you can see that we will be able to take care of the document and also understand whether it's gonna to be any of the good things. But you need to understand the coefficient of the variation, whether it was being working to the CV or not. For example, in one lab, and actually it's the CV monthly record, but it is not meeting the ultimate criteria. Still, it will be taken as less qualified. So many people believe that after identifying the gap, what we can do to that. If it is a CV is not meeting the criteria for that month, what can we do? You can say that for some of the labs, actually, their failed rate is less than 5%, only 3.9%. Then we need to think about, should we actually take measures to further improve our cutoff achievements? And then for this lab, I believe we need to do so, whether 5% of the failed rate or the coefficient of variation failed rate can actually help to lead to further improvement. CVs being not meeting the quality indication, there are a few reasons. First of all, maybe the quality management target is being set at too high. As long as your practice is compliant with the clinical needs, you can surely begin to elevate your quality management target, methodology, and also the environmental factors means a lot. For example, for the testing system, whether you have the expired reagents, whether there's any kind of sample contamination, and also there are some of the factors that are going to impact the ultimate quality of the testing. On this table, I also shows there are some of the quality control average rate that started to drift. If you do have some drift, then surely speaking, your test results will not yet be meeting the quality indication. You can say that for this lab, its calcium CV is 2.0. So we need to talk to the manufacturer to say whether calibration period really works for the instrument label. Should we shorten the calibration period or something? And we should also take improvement. We need to pay attention to the CV to see whether it actually meeting the criteria. If we believe our project CV, you know that you have to find the evidence to choose the best methodology. As long as you meet the criteria for CV, you will be able to leverage the best testing methodology. In that way, we need to actually make sure that we have a good quality indication comply rate of more than 100%. If there's some months that you fail to reach a target, you have to find the countermeasures for improvement. If your monthly CV is not reaching the quality indication, you need to find the reason, identify the gap, and then to take the measures to make sure your annual CV is actually meeting the quality indication. And you mean that we have a so-called failed rate. Failed rate doesn't mean that the CV could be subbarred. In the year of 2017, and in the document, it started to have the new indications. We should also be more prioritized on the quality indication, monthly review, and analysis. your lab's been operated for so much long a time, you have to make attention to the analysis average rate and X. For example, each of the reagents or each new batch of the reagents, and you need to understand its average value. And also you need to understand whether the CV can actually meet the quality indication. You should also understand whether the system is capable of providing such accurate rating. You need to understand what would be the frequency of the failed operation. If there's more frequencies identified for any of the failed case, you need to identify the root cause. I made a sentence in the bottom of this slide. When needed, you should actually help to modify the average value. But personally speaking, you need to be cautious because average rate in the lab needs to be maintained. And it should be maintained and should be a stable number. It might have some numeric changes, but averagely speaking, average value of the lab need to be well maintained. This is actually a point I'd like to mention again and again. And don't just easily to modify any of the quality criteria in your lab. And the second part is WBC. Case regarding how your project CV can go beyond the quality target and the quality requirement. You can see that on the table, I show the precision that the coefficient of variance for WBC need to be less or equal to 6%. If you make this number 5%, then that would be great. And you have to make sure that after long-term operation, maybe your long-term target is to make a coefficient of variance CV less than 3%. So you can say that for our CV, for WBC, it need to be at least 5% according to the government regulation. But actually in your practice, you need to strive for the best, maybe make it 3%. If you make it 3% every month, then you will be able to see how stable it's been operated. And another point is that the failed rate. And for the lab, and you need to make sure that the pass rate or the need to be more than 98%. For majority of the labs, it's more than 100%. For example, for this creatinine project, and it actually started to have some negative variation, it's 7%. So we have to take some preventive method, because you can say that maybe there are some major changes to the average value. Then we need to do the root cause analysis. And we need to, first of all, trace back to the track record of the average value change within the lab. You should also benchmark with any other centers in your same province. And also taking a look at the instrument, the calibration, and the batch of the reagents. Then you can take some measures in order to be preventive for any of the negative variation. And for sure, we can identify the root cause. And then at the right time, when you have enough evidence, if you have a negative variation, then you can actually start it to modify the criteria you have. You can see that for this project, it actually targeting the bilirubin, and actually it's a bilirubin PT has been failed. And how we're going to do the quality analysis. You know that for this lab, they did the third PT in September 2019, still it failed. And you can see that right after May, right after June, whether the average rate of the lab has been changed a lot for the bilirubin. And we should also consider the quality control situation or landscape in the same lab on the testing day, making sure that the testing instrument and system is robust. Otherwise, it's going to hurt the test result of the patient. So after the data analysis, and we identify, and when we have the bilirubin project, and there's just one sample that is being placed for a much longer time. And especially for the redialysis time, it's 165 minutes and much longer than the 30 minutes requirement. And also the environmental factors impacted the ultimate result. So that's the reason targeting the problems to be identified. Then we can take the corresponding measures for improvement. The corrective measures include the following ones. First of all, you have to follow the SOP, making sure that the sample, its redialysis time should be less than 30 minutes. But in the first test, it makes 165 minutes. And right after the measures being taken, we should also understand whether then you know that after the improvement measure, the second test being perfectly passed with all the quality indications being met. And the result is being nicely presented. Where at the same time, we should also consider how it's going to impact the patient result. I have to mention it again. This is a very important point I'd like to draw your attention to. For my presentation. I also welcome your comment.
Thank you very much, Professor Xiong, for your fantastic lecture. As we know, the ISO 15189, the new edition, is planned to be released this year. For the new edition, some of the revisions are around the focus put on effectiveness of the management and reduce the risks for patients. The quality indicators help labs to quantify the risks in the process of the workflow of the laboratory and provide the supervision and the surveillance. So at our work, the drafting of the quality indicators, it is not perfect yet. There are still issues to be addressed. Just like Professor Xiong introduced, each lab has its own characteristics and its own quality principles and quality goals. Then you need to actually set up those contents which are specific to your lab, their quality requirements and the industry standards, as well as the applications and the continuous improvement. And a focus on the CV value for internal QC, and also, for example, conformity rates based on external quality control results. So I'm sure we've learned a lot. I'm sure it is of a great value to our future practice. So now let's thank Prof. Xiong again. The next part, we're going to hand over the moderatorship to the next expert, Professor Feng Ning. Prof. Feng is the Qianxinan People Hospital and the chief of a lab testing department, Guizhou Province, of the Anti-Cancer Association Tumor Biomarker Committee Standing Committee member, and a Guizhou, the Integrated Medicine Committee, a standing member, and a Guizhou Medical Society, the Lab Testing Branch Committee member, and also the Guizhou, for example, a Reviewer Committee expert, and a Guizhou Southwest Professional Technical Assessment Committee member, and published 20 papers, and chaired those research projects, and awarded with 2 first prizes of a science and technology progress. So without further ado, please let me pass the floor to you, Prof. Feng, to moderate the next talk. The next speaker is Professor Wan Nan, and a PhD, MD, and a Master Advisor, published 30 SCI papers, and chaired 1 national key research project, and also the COVID research project, Liaoning Province, a key R&D, and a natural fund projects. So his research directions are around pathogen molecular testing. Right now, he's also in charge of the lab quality management and clinical chemistry work. And he wears multiple academic titles. For example, the Deputy Committee Chief of the Expert Committee for China Integrated Lab Testing Platform, and also Liaoning Province Immunological Society, the Youth Committee member, Deputy Chief of this committee, amongst a host of other academic hats. So Prof. 1 comes from China PRA, North Army Theater General Hospital. The title of the talk is IQCP Practices. Over to you, Prof. 1. Thank you very much, Professor Feng, for your introduction. It's my pleasure to have this opportunity to come to the beautiful city of Chengdu, which is my hometown, by the way, and deliver a presentation about IQCP. In terms of the contents today, 4 parts. So actually, some of the contents are overlapping with those already mentioned by Professor Nichols. IQCP, the journey of the development of IQCP, the concept, and also the basic process for IQCP. These are some of the 3 parts of my talk. Back then, my experience with IQCP go back to 2015. I went over to the United States, Atlanta, to join in the AACC annual conference, which was the largest annual event of our lab testing area. So after registration, from a website, we were able to download the profiles of those speakers. So there is a whole host of breakout rooms, and I actually heard a presentation centered around the topic of IQCP. The speaker is actually, for example, the chief of the Joint Commission. And in 2016, IQCP was about to be rolled out. So in 2015, the very concept was presented during that event. So I thought this topic was very interesting, and I wanted to carry out POCT for blood gas testing at my lab. But we didn't succeed at that effort. But luckily, this time around, thanks to my reorganized forum, so I'm here to deliver a presentation about this topic, which I deem as very interesting. And what about the IQCP journey and its evolution? So actually, the testing technologies are evolving continuously. A whole host of manufacturers built in the sensors in the devices. As a result, the devices are more robust. There is very good reusability of the results. So some labs put forward the concept that they do not want to always follow the clear requirements. For example, QC at 2 levels every day, which they think of as a waste of time and less efficient. So they do not want to follow the clear path. Therefore, in 2013, there is like equivalent QC concept, or EQC in short. So, according to Annex 3 of a CLEAR, some labs are allowed to follow this alternative path. But in 2013, CMS, as Professor Nichols mentioned, included EP3 in CLEAR IG. After that, it was found those EQS projects, there was no information exchange opportunity with clinical labs. So there was no loop closed for QC. They only monitored some of those intra-testing risks But what about the sample quality before the testing? There was no surveillance there. Therefore, it didn't cover all those risks and variables throughout the whole process. Therefore, in 2015, CMS put forward the very concept of individualized quality control plan, or IQCP, and then repealed the concept of EQC. For CMS, a brief introduction. It plays a role which is similar to the role played by China National Health Commission. It takes care of the clinical lab administrative and financial management of American clinical labs. There is also CRSI, which is a standard institute, which is similar to the TD committee in China. So CMS takes care of the management of the labs. So CMS is the most important institution. Hence, it puts forward those very important standards, like EP3 and IQCP. Then the basic concept and the purpose of an IQCP. So this concept originates from foreign countries. So let's look at the original definition. The keywords are marked in red. It is an all-inclusive approach. It covers every step of the process, including before, during, and after the testing. Second, applied to all non-waived testing performed, including existing and new test systems. So it is not only about the POCT devices. For existing initiatives following the clear two levels path, you can also refer to IQCP in your management on a voluntary basis. So the labs are now have flexible choices. So at least 5 parts shall be included for IQCP. Like Professor Nichols talking about molecular testing, for sample, equipment, environment, reagent, and personnel. The 5 parts must be included in the assessment. Then the QC frequency and the level shall be determined as per risk assessment. Risk assessment is fundamental for IQCP. For all testing processes, you need to complete a risk assessment before you're able to come up with a QC plan. And moreover, IQCP is not overlapping at all with CLIA, but rather it is a very good supplement to it. It is based on voluntary basis with respect to whether you use it or not. Third part of my talk, the establishment process for IQCP. Pretty straightforward, 3 parts: risk assessment, then quality control plan, then last but not least, quality assessment to analyze the completion status. For our quality management work, a very important concept is closed loop management. So no matter for 15189 or some other concept, the closed loop management is very important. It is through risk assessment that we evaluate the source of errors in our testing and which are not acceptable. Based on the assessment results, then we proceed with the QCP planning. In the planning, we need to clarify the acceptable criteria. We need to tell whether it is good or bad. We need to be able to identify the errors. This is the very purpose of the planning. Moreover, for the results of the planning, we need to check whether they are valid. For instance, quality indicators, good or bad, high or low. Yet we need to carry out assessment. If the result is not good, we need to carry out corrective actions. From that perspective, we can close the loop on quality management.
This is actually the process of IQCP. Ultimately, we'd like to build the quality culture within the lab. This is our fundamental target. We're not there for certification only. We'd like to build a robust system for quality management within the lab.
这张图呢，我觉得是一个
This is actually a typical procedure for IQCP. I personally use it a lot in my lab. This is actually the procedure shows how the IQCP should be constructed and what need to be done. First of all, from the inputs perspective, we have the samples, instrument, reagents, testing methodology, staff, operator, and continuous education. As long as we have inputs, we have to have controls because we are in a closed loop environment. For example, what would be the criteria in accepting the sample? What would be the criteria for rejecting? And how should we maintain the instruments? And how should we keep the instruments? For each one input, we can have 1 to 2 or even 3 corresponding control measures. It is not a linear 1-to-1 ratio, where during the testing process, how we're going to monitor the whole process. We need to have the quality indications be set in advance, because ultimately it can help to decide the testing efficiency. And it doesn't mean that the higher the quality indication be, the better the result would be. Otherwise, it's going to interrupt your work efficiency. You need to find the comfortable level of the quality indication. Otherwise, if you set the bar too high, it's not going to support your daily work. During the process, we should also do very good control, as I mentioned. We do have the testing, review, benchmarking, and talking with clinicians and getting the clinical appearance. Ultimately, we need to provide the outputs, including the test data, test result. You can say that by building all those testing data into one report, we can then go back to see whether our testing is being properly done and whether we do frequent control and whether the measures for controls being well implemented. So you can say that in the process of control or in the process of testing, you can see we need to do documentation and control. In other words, and we need to build the institutional mechanism for documentation and control. We can also take internal audit to make sure that all the reports is being properly generated or produced. And ultimately, we can take management review to help to adjust our internal process and then to take corrective measures if there's any need. We're talking about risk assessment in IQCP. We have to consider EP3, a typical reference document. This is actually the sequence of events creating risk of harm for a patient. In EP3, it's being broken down into 6 steps. Let me just introduce them one by one. For P1 and P2, and that is initiating course testing process failure, and P3 were after the testing, and the P4 to P6 are actually what's going to happen in the clinical setting, especially in the diagnosis and the treatment process. So these are the typical 6 steps. It actually helps to build the sequence of the events creating risks for harm for a patient. And there's a typical formula being set here. That is a probability of harm equals to P multiple E and divide MPBF. Due to the time reason, I'm not going to elaborate on this formula. Just would like to briefly introduce MPBF is actually the time or the sample size. It could actually be generated. For example, you can actually take a look at the risk management and the statistical QC, and also you need to consider how many, for example, samples you are going to actually test per day, and whether there's any errors being generated. This is actually the that could be every day. PHU is actually the clinical harms being generated in clinical setting for the patient. It cannot yet be quantified, but it's highly relevant to our testing result. For example, like some of the bio, for example, like troponin, and if you're going to find the wrong number for the troponin, and it's going to impact or jeopardize the whole testing process. So you know that we have to identify some of the key testing items. And we also have ENUF, the green labeled area. Every day when you do QC, you have to take corrective measures, but corrective measures can only be used for the previous testing. But you know that for our lab quality indication, it's more like the sampling and then to generate, identify the gap. For some of the uncorrective measure and the ENUF, we're going to represent the for example, the false data or the wrong data. If you can identify those problems earlier possible, then that can help you to identify the corrective measures more accurately. So we can control PHU and also MPBF. And ENUF and MPBF are actually the two ones that we can really control in order to mitigate the harm to the patients. Let me just use a flow chart in showing the risk assessment. I just list it out here on the slides from sample collection and to the appearance to the clinicians. The most important control point is that how we're going to do the monitoring of the risks. What would be the methodology? This is actually in line with IQCP process. We need to have a whole journey risk assessment. And later I'm going to give you some concrete cases to share what we can do with that. Well, talking about risk assessment in CLIA, there's actually a EP3 templates. You can download it from CLIA website, including 5 steps to go. It's different from FCD. For example, it doesn't mention about the staffing, but it's already been included into instrument or reagent. If you'd like to do the risk assessment over your instrument in your lab, you can use EP3 templates to do one-by-one assessment. This is actually a template with great granularities, for example. And for sample collection, we actually have multiple breakdowns being built into sample collection part. If you'd like to take a look at it, I suggest you to go to the official website of CLIA to download this template. Well, the second step is to build our quality control program or plan. The most important thing is to make sure you need to identify error. Where can you actually identify those errors that's going to generate high risks? How can we identify the error? That's the most important thing. We have to build a QCP in order to identify those errors. The content are the 5 elements for assessment, but as I mentioned before, but it's not yet being limited to that. If you'd like to have something more, you can also build your own criteria inside. Because for QCP, ultimately, being identifiable is the most important one. So your quality indication need to be well set, being stratified, and also making sure that your quality indication need to in line with regulator requirement and also manufacturer requirement. And also the lab head needed to confirm and approve the QCP in order to make sure it could be well executed in the lab. The final part is quality assessment. That is QA. It's just like a review process. It actually builds a corresponding procedure and measures, taking corrective measures, especially monitoring and assess whether the corrective measures are valid or not. We need to assess that. According to the assessment result, we're going to see whether do we need to adjust the IQCP. We always say quality assessment and quality control, what's the difference between the two? Quality control is more like a behavior. For example, the humidity temperature in the lab, the temperature of the refrigerator, and the training over the staff. You can say these are actually the so-called quality control, where for quality assessment is to review the data generated from a quality control process. So quality assessment and quality control are different from each other. Generally speaking, QA is more like the analysis, where QC is more like data collection and control process. When we build IQCP, the process is pretty easy to go. It's not that complex. I think maybe in our daily life, more or less, we have already touched upon IQCP. Even the quality indication and production also involves IQCP. But we haven't mentioned that too much. I think IQCP, more or less, as all of you sitting here, have already taken it as a part of your daily practice. If we're going to build IQCP in our lab, there are a few points we need to pay attention to. IQCP is being built upon the harms or the risk source. If you have a multiple instrument in different locations, they're going to have a different source of the risks. So you have to have IQCP for each location. And even if it is the same testing equipment, but with different testing module, you should also have IQCP for each of the testing module. And also, CMS haven't made any regulation on which program need to use IQCP. But if any of the lab failed to reach the lowest quality control requirement made by CLEAR, then it need to adopt IQCP as a compulsory need. For example, any project involved with the microbiome, molecular testing, or the POCP, or any of the project that used to adopt EQC, they all need to go through IQCP mandatorily if they haven't met CLEAR lowest quality control requirement. That's the regulation from the US. So why should we mention IQCP here today? First of all, you know that in domestic China, and we already have the stratified medical consultation. You know that for hospital at a different level, and we also have different labs with different instruments and service being provided. But we need to be more flexible in adjusting our quality control process so as to make sure that the quality management could be in position. IQCP can allow our labs to be more flexible in using the quality control measures by complying with the laws and regulations. And more importantly, we'd like to emphasize on risk management and closed-loop management so that we can generate a continuous improvement to really meet the target of IQCP. Risk assessment tells us where does the source of the error come from, which steps is going to be compromised. QCP can help to answer when can we identify and prevent error in a timely manner. QCP is based upon risk assessment. And the final part, quality assessment, is to say whether the IQCP that is being implemented is valid or not. Okay, talking about some other resources, you can say that on CMS website, there are so many IQCP guidelines, and also be able to provide you the training session if you'd like to learn more, and I'd like to suggest you to go to CMS website to learn more about it. They do have the step-to-step instructions. Okay, let me go to the fourth part of my presentation. That is IQCP practice. Because IQCP is actually being done in the US. So how the Chinese lab can adopt IQCP? First of all, we need to understand the lab management method in China, because that is the fundamental framework of the regulation we need to draw upon. You know that from the regulation perspective in China, and we have a medical institution clinical lab management method, which has been released in 2006.
Then there is GB or industry standard, or others, those guidelines published, for example, 2008, which is a domestic GB, which was converted to GB. And there is 22576 2018 edition. Right now, there is accreditation rules published by the Commission in 2013, which is the most adapted. Then regarding lab QC, for example, there is GBT 20468, referring to the American guideline. So for internal QC requirements, these are the laws and regulations and standards in China. It is stipulated at least one test shall be performed on the controls for every analytical batch. Then how shall we proceed with our efforts at our lab if the minimum requirements cannot be met? Then we need to be very careful. The IQCP process is very useful. So this is my lab. You can look at immuno group in a chemistry group. The QC rules are implemented. But for some other teams, the internal QC efforts have yet to start due to various reasons. For these groups, if the industry standard cannot be met, then we pick some projects from these teams on which we carry out IQCP QC. For example, chemistry group, you can look at the urine testing items. The samples are very special. The microalbumin in the urine and creatinine. For these couple of items, we established IQCP. So first, we look at the label instructions, whether there are any specific requirements for QC. So the first, this instructions manual that we read first and foremost. But after that, we perform RA, risk assessment, which includes at least 5 aspects for this very project. Taking sample as an example, which is similar to what we heard from the American expert, including sample transportation. Since it is urine, it may break or it may be lost. As a result, the sample may not be up to the requirement, and a barcode for the sample. And we have the columns that say the probability of occurrence and the severity and the level of acceptance, etc. Then different sources of the risks, including the devices and the reagent, etc. So one of the possible sources of the risks is a collection of the samples. Another possible source is the devices, the environment, and the personnel. The 5 aspects that we cover in our analysis. Then we find out about those risks which we deem as intolerable. So those items we need to list out on a list. Then we talk about the monitoring frequency, daily, weekly, or monthly. We need to fix a frequency according to which we perform those efforts on a regular basis. Then we need to be up to those standards, which are able to meet the standards, which fail to meet them. Then assessment. We have the documents, tables, and the data, the notes, the temperatures, and etc. For one source of the risks, we may monitor 2 or 3 points. That after we assess the entire documentations, then we output the new quality plan. In summary, the basic framework for establishment of IQCP. The 5 parts. The latter 3 are more important. First part, we need to clarify the quality managerial goals. For instance, like the internal QC. And there are complaints from clinicians. If there is a lot of complaints that we hear, that it is necessary for us to monitor this very item. And who is the owner of these efforts, the time it takes. That in terms of a risk assessment preparatory stage, we need to collect the data. For example, the competence, the temperature, humidity, instruction label, and also performance verification, etc. And based on this data, then we carry out the assessment, the RA, including pre-testing, during testing, and post-testing. Then we should cover the 5 aspects, such as sample, equipment, reagent, etc. Then QCP and a QA. So this is actually the basic workflow for IQCP. At my lab, it's not like this is mandatory, but at least this is like a novel kind of a mindset that we have for quality control. Identify the weakness in the process. For some hospitals, it's very difficult for them to establish a full-blown QC system, but you need to focus on those priorities. So I think IQCP is definitely of practical value. Going forward, if you apply it in your practice, I'm sure you're going to accrue your experiences and you're going to do it in a better way going forward. For 15189, throughout the years, I think innovation is of the utmost importance in the quality management work we do. For example, 15189 talks about whether it is suitable with you or not. Yet you need to innovate based on the basic framework of 15189. You need to apply them in your practice, just like a pair of shoes. If you only buy the pair without wearing it, I don't think you're going to have a feeling about it. Then the IQCP, you need to apply it. So these are some of the literature that we refer to. For example, 20468, 2006, and also WST 641, 2018. Thank you. Thank you, Prof. Wan, for a brilliant talk. Professor Wan talked about the IQCP, talking about the risk assessment for labs, the QCP planning, and also to close the loop on the entire managerial process. He talked about in detail the risk assessment under IQCP, 3 phases, 6 aspects, some of the caveats. So as far as IQCP practices and concept are concerned, when it comes to the primary care institutions, I'm sure it is of a great value. We aim to meet those quality requirements to deliver a certain performance level. So it may entail different QC strategies for different projects. it's necessary to establish this QC strategy for each project. So thank you very much, Prof. Wan. And back to you, Professor Xiong. Yeah, first, I'd like to thank you very much for your moderatorship and Prof. Wan for your lecture. With that, we heard 3 talks talking about individualized QC. When it comes to QC, the quality indicators, quality goals, the application of them, and the continuous improvement. And we've heard comprehensive yet practical knowledge and information. We also have 3 moderators who chaired the 3 sessions and passed their comments. So great presentations. And based on the contents that we heard today, and those elements about QC, we definitely look forward to more exchanges down the road. The next, let's pass the floor to Conference Chair Professor Xiong for the next section. Thank you, Prof. Xiao. As of now, we've heard 3 talks. Professor Nichols, Prof. Wan, and we heard these presentations. So I'm sure from the main room and the breakout rooms, we may have some questions for our speakers. So from a Guiyang breakout room, do you have any questions from your side?
We're
from Guizhou breakout room. We're from the lab testing department, Guizhou University of TCM. Thank you, Professor Xiao. And thanks to Professor Xiong for inviting me over. Thanks to our experts for their fantastic lectures. I've got a question here for Professor James Nichols. Honorable Prof, first, thank you very much for your sharing about EP3-A in terms of the application of practice at medical lab. I have a question for you. For POCT, QC, quality control is always a challenge because just like you mentioned, for POCT, most of the POCT are carried out by non-lab professionals. For these non-laboratory professionals, they may not understand the QC rules. They tend to ignore some of those steps or 5 aspects which have a bearing on QC. They may ignore those important factors. So my question for you is, without compromising patient safety, how can we strike a balance between the cost of QC and the effectiveness and outcome of POCT? Prof. Nichols?
So I'm going to come back. Can you hear me okay? Yes? Okay.
Yes.
I'm going to come back to the IQCP because essentially, when you make an IQCP, you're considering those 5 factors plus everything in that setting. And the setting that we're dealing with is non-laboratory operators. These are people who minimal laboratory experience. They're thrown into this because of their clinical responsibilities, and they don't necessarily want to do this. We have to realize that as well. They want to take care of patients. That's what they were trained for. They don't want to take care of lab testing. They don't want to do quality control. They don't want to maintain equipment. So we have to factor that into our risk assessment. Here is a unique setting where we're dealing with people who don't necessarily understand laboratory testing. What extra steps do we need to do to bring them up to the level of what we consider quality testing? maybe additional training, maybe components of the training that actually focus on some of the aspects of pre-analytic, analytic, sort of post-analytic variation, and particularly those with sample collection and analysis. And that's why I focused on molecular. You know, we were very concerned moving molecular to the point of care, and we added additional quality control that was not part of anyone's manufacturer, anyone else's program. We said for moving molecular COVID testing into a point of care environment, we're going to require that the point of care operators, one of them, whoever, actually weekly test the environment, meaning they're going to take a swab, they're going to dip it in viral transport media, and they're going to swab the countertops, the analyzer, the keypad on the analyzer, the doorknobs on the room, wherever else, and then they're going to test it like a real patient. If it turns positive, we know that they have not done what we've trained them to do, which we took extra precautions in training in moving molecular to the point of care. We said there is high probability they're going to cross-contaminate these samples. So we want them to handle one sample at a time. We want them to change gloves. We want them to clean between samples with bleach and make sure that things are clean before they go on. If they're not doing that, it's going to show up in the environmental control. So that's sort of our risk assessment of what could happen. And there's our mitigation steps of what we could do as control processes to prevent that from happening. That went into our quality control plan. And you know what happened as we assessed this? Not a single one of our clinics actually had a positive environmental control. We rolled it back to monthly now. We just kind of want to keep pace with it and make sure that it's staying par. But the problem we had was our emergency department. You know, this is an area where they are trauma patients. They're not taking a lot of care. They're like rushed. And we got multiple environmental positive controls, meaning they weren't really following the policy we had trained them to. So we stopped, we sort of reassessed, we went back in, retrained these staff, and they're doing much better now. So it just keeps, you know, it is a cycle. It is a continuous quality improvement cycle. And we need to keep reassessing where are we having problems. Let's target that area and let's go in. And this is what IQCPs really help us do, risk assessment in a more general umbrella. That's what helps us pinpoint where our problems are, and let's focus on those. So I hope that answers your question. Thank you.
Thank you very much. Thanks for Professor Nicholas. Thanks for Professor Xiong. That's my question. Thank you. Thank you. Thanks for Professor Nicholas. Very good response. And I surely believe from the answers being proposed by Professor Nicholas, we know much better than I think Professor Zhao from the Chengdu venue may have a few questions. I'd like to welcome Professor Zhao to raise a question. Distinguished Professor James Nicholas, good morning or good evening to you. I have a question here. When we do the assessment, especially risk assessment, you know, sometimes there are three major factors we need to consider. First of all, that is the risk validation. Second one is severities. And thirdly is how likely the risk going to happen. You know that in EP3 and also even in ISO15189, you know that some of the so-called risk assessment criteria is being clearly listed in the document. But you know that when I was listening to a presentation, I find out in the new EP20 document is going to be available. In that available, and the risk assessment will be built into the, will be more kind of refined. So I'd like to know that for EP23, because it's been rolled out for many years, maybe we're not going to have the revised version. So for labs, when we are benchmarking the EP23 for our practice, maybe there are going to be some is there going to be any new contradictories between the new version and the old version? Do you have any good suggestion to the labs? How can we consider to switch from the old ones into the new ones? I mean, the EP23. Thank you.
No, there will be no conflicts between the old version and the new version. There'll just be additional information to help you really create a good IQCP. I can say that we purposefully left detectability off of the previous document as sort of an assessment of your prioritization of what you're going to address first versus second versus third. Understand that the numbers and what's in ISO risk assessment, when they talked about detectability, they talked about severity of risk. These are all factors to help you as a laboratory assess what is your greatest priority. What do you want to address first? And what do you want to address second? And those numbers are very fluid. They're very independent on the lab, the hospital setting, whatever. They are not set in concrete. They're not part of the whole IQCP process, other than the fact of helping you prioritize resources. We are all very short-staffed. We don't have a lot of resources to expend to just philosophizing about like quality control. So this was an attempt to help you just sort of rationalize, hey, you know what? This is our greatest risk right now. We need to address that top priority. And if you look at the lower numbers, oh, look, this is our second priority. And look, this is our third priority. And this is our fourth or fifth. And this is really sort of way down there. And that's why that the numerical system actually even entered into EP23. For me, I think you have some common sense. You kind of know what your top priorities are. You could throw out all those number systems and just say, hey, I want to address this first. I want to address that second, and go for it. You know, that's what the beauty is of like the risk assessment and the IQCP process. This is, we tried to provide tools. That is just one tool. So hopefully that helps. And it's not going to conflict with previous editions at all. It's adding additional numbers. People were kind of going, how do I factor in that the manufacturer has this, that, the other? Well, we'll add detectability to those number systems, and it doesn't really conflict with what you're currently doing. Hope that helps.
Thank you very much. Thanks for Professor James Nicholas. Very good. So we really look forward to see the new version and hope the new EP23 would be available as soon as possible so that we can build it into our lab practice. Thank you.
Me too. We're working on it. It's in the voting process right now.
Thank you. Thanks for Professor James Nicholas. Coming next, I'd like to pass on the floor to Professor Chai from Kunming subsession. Professor Chai, do you have any question you'd like to raise here? Hello, testing. Yes, we can hear you. Great. Yes. Thank you very much. Thanks for Professor Xiong of having me here. We are now in Yunnan subsession in Kunming. I come from the third affiliated hospital of Yunnan TCM University. I have a question to Professor Xiong. Professor Xiong, good morning. You know that in our daily lab practice, how can we actually monitor the quality control indication to see which process is more risky, and then we can set up our own IQCP. Thank you very much. Thanks for Professor Chai. This is a question for me. So actually, talking about the quality indication, I think the key is still on coefficient of variation, that is CV. But we not only need to keep an eye on CV, more importantly, we need to leverage the daily statistics or some of the average value changes of the lab. CV is quite important, but more importantly, we need to understand the reason why some of those ratings are not valid. You should also pay attention to the average value of the lab. And also, you need to make sure that you have a good calibration system in place in the lab. You need to have a good calibration process being done in your lab. And after you find the right testing system, the batch of the reagent should also be considered because you're going to use a new batch of the reagents. Probably the test average value of your lab are going to be changed. For a coefficient of a variation, if it reach certain level, we're also going to say why the CV is being changed. If you change a certain testing methodology, which is the poorly performed one, then CV going to tell you the testing methodology is not yet be well chosen. And you should also make sure that rigid reagent management and batch stability of the reagent need to be well controlled. If CV is being compromised, maybe we can improve the frequency for quality control or even improve the quality indication. By taking all those measures, we'll be able to further improve the quality control process in the lab. Thank you. Thanks for Professor Chai. That's my answer. Thank you, Professor Xiong. Thanks for your nice response. For sure, in our daily practice, especially in our lab process, we're going to pay more attention to calibration, the average value, and also sometimes improve the frequency of the web quality control in the lab. So hope that in the near future, we're going to have a well-established IQCP. Thank you. Thanks for Professor Chai. Thanks for your question. So let's see. Is there any further question from our audience, our Chinese colleagues? If you have, we still have a few minutes that you can raise your question. Professor Xiong, sorry, can you hear me? I'm Professor Deng. Thank you, Professor Xiong. Thank you very much.
Thanks for Professor Leko. Our side has two questions. First question is from my colleagues. So I will send the mic to my colleagues. Okay.
Hi, Professor Nicholas. I come from Chongqing. Thank you very much. And thanks for Professor Nicholas in sharing with us POCT risk management. I do have a question that is different from risk management, but it is also a key part for quality management. It is more on the managerial side. Professor Nicholas and all the Chinese audience, I'd like to ask you one question. You know that for each hospital, when we do POCT management, does every center have a management committee or the management task force to POCT, making sure that we have a MDT team to do the management of the POCT, where the tasks being split over between the central labs, the management team of the hospital, and the clinical side? You know that we are not involved in the reagents selection or the instrument selection. Then the hospital asks the lab to manage everything. It's very difficult for us. What's your practice at your center? This is my first question. I have another question. I do hope that we can actually ask the question to other Chinese colleagues. You know that in my hospital or in my district, I received patient complaints about a POCT. The local healthcare commission asked me to read through the POCT guidelines. There's a glucose guidelines being specifically made in China and also an expert consensus. For the administrative department of each center, do we actually have a permit or certificate ask the nurse to go through or ask them to receive the education? Otherwise, you know that for each lab, it's very hard for us to manage all those nurses because they are not yet being certified or receive the uniformed training. Thank you.
Sure.
Yeah, for James, from your country, are there any management model we can learn about? And other laboratory experts from China as well, you may share with us how you manage your hospital POCT. How do you allocate those performance?
Sure. So I think we had several questions there. Let me kind of take them one by one. In terms of the way that we manage our program, we have a point of care staff that is laboratory owned. They're paid by laboratory account out of the hospital, but they are run by a point of care coordinator who's the manager. And they have staff that help in terms of training, competency, issues with new test requests coming from the floors. What happened recently is that we've acquired a lot of other hospitals. So now we're an enterprise, multiple hospitals and multiple staff at different hospitals. And we brought them under the same umbrella. So common policies, common training, common competency tests on an annual basis, just to make sure that staff are keeping up with knowing how to do this. If they do it very infrequently, they'll forget how to do a glucose test or a urine pregnancy. So you have to maintain competency. So that's sort of the umbrella that we brought them under. Get them all uniform, get them under the same policies, training, competency, and management structure. And they are managed by their own staff. They're nursing. They report to nurse managers. We are laboratory. We're point of care, but we can consult with the nursing staff and say, this is the best practice. This is how you should be doing things. We can help go out on the unit. We can inspect, make sure they're taking temperatures, checking in reagents, maintaining their reagents and throwing out expired reagents, and the environment's clean. There's not blood everywhere, urine everywhere, that it is a safe practice. So I would say that is our goal in terms of point of care practice. And that in the United States, I would say is pretty much what we want as standard across the institutions. Every institution will be a little different, but within an institution, you have a lot of different nursing units. You may have outpatient clinics that fall under the umbrella of the hospital. And we have, I want to say, 150, 200 clinics that are affiliated with the umbrella of Vanderbilt. Everyone shares the same policy, same training, same competency, and same goals in terms of doing this. So getting everyone on the same platform is, I think, number one to quality. Number two, what else can we do to improve quality? I would say that it's really emphasizing pre-analytic and analytic following policy, making sure that they're doing what they're doing. I would say the complexities we're having right now that I'm facing is regarding requests by nursing because of nursing shortages, staffing shortages. They want to delegate the responsibility of point of care testing to lower and lower education staff. Staff that are in the United States, they are high school diplomaed individuals. These are people who are no medical background. They may know glucose testing from their family if they're diabetic or whatever, but they want to delegate the responsibility of doing glucose testing to people with no medical background. In essence, no education. They've been trained on how to run the meter, but that's about it. They don't know what the glucose test means. They don't know how to treat the patient. They don't know what the treatment would be with this. And so here's a setting where you have to worry about, does the patient have good peripheral circulation? Is a capillary finger stick really relevant? Should you be doing a capillary finger stick on this particular patient? And that's a medical assessment that this particular individual really can't make because they don't have that background. They don't have that medical education. That's a nurse, a nursing staff type of setting. So it's the conflict that I'm too busy as a nurse to take care of all this laboratory testing. I want to delegate it to someone else to do the testing. But we, as a goal of the hospital, the laboratory, want uniform patient care, no matter where you are, no matter by who's doing it. So we have to ensure from the laboratory that these lower educated people, I'm not saying lower educated, they are lower experienced with the point of care environment, and they really can't take on the medical assessment of peripheral circulation. This is a problem for them. So I kind of throw that out, that this is a complexity of the point of care environment that we're all thrown into once we take the lab out of the lab and take it out onto the floor. And that's why we do risk assessment. Every setting out there has certain risks, certain differences that I may say, okay, it's okay for this patient care technician or this care partner to do glucose testing in a general medical unit in my hospital, but I don't want them doing it in an intensive care unit with a critically ill patient who may not have good peripheral circulation, and a capillary finger stick isn't a good sample for any point of care, not just glucose. I hope that sort of addresses some of the questions you were asking about, risk, how we deal with point of care training, these types of issues. Hope that helps.
Thank you, James. So in your country, you have a platform to provide the uniform management of those staff. Yeah, thank you. Good experience. Thank you.
Okay. Thank you, Professor Lix. We also know that EP3 guideline is a useful tool not only for central labs but also the POC unit. But I think there is something different from American and China. So we want to invite you to China and customized regulations for Chinese hospitals. So we look for that. Thank you. Thank you.
Thank you. I look forward to visiting again. It's been several years since I've been in China. I may have met several of you when I was in China, Hong Kong, Shenzhen, Guangzhou, Nanning. I look forward to meeting you again in face-to-face person. Yes.
Great, thank you. And thanks to our guests from Chongqing breakout room, and also thanks to Professor Nichols for a great answer. Thanks. From Chengdu breakout room, any further questions? We may still have some time to take more questions. This is a great opportunity. But if no, as of now, you've been working hard. Today is the weekend. We'd like to say a big thank you to all the experts, especially those participants who are here on site in Chengdu and in Chongqing and in Kunming, Guizhou, and Guangxi. Thank you very much for taking time off your schedule. We'd also like to extend our particular thanks to Professor Nichols from the United States. Thank you for your wonderful lecture and your answers. We look forward to the face-to-face visit of you to China. And also thank you, Prof. Wan, of your lecture on site. And also thanks to MindArray for the great support, the platform it has created for our exchanges. So our theme today is the risk management-based individualized quality control. I think this is a very relevant theme, and we're honored to have with us the top experts from the world and from China. So this is a great opportunity. In the panel discussion session, and we had discussions on a whole host of topics or questions. It's our great privilege to have today's opportunity for mutual exchanges. So in the interest of time, with that, let's wrap it up. Thank you again. Thank you all.